So this was great. And you'll find, thank you for this generous introduction. But there's so much, Mitch's talk resonates so wonderfully. It's my own experience, really finding something. I don't think I've ever worked a single day in my life. Just coming in and doing what we do. And what I decided to do today is give you a little vignette into what keeps us so fascinated about our work. And basically, we are not engineering things, but we are detectives, right? We are explorers. We're trying to figure out how cells work, how organisms work, and are put together and functions in so wonderful ways. So if you think about it, our bodies are really inert. They contain 10 to the 13 cells. That's a lot of cells. It's about world population is 7.5 times 10 billion, 7 billion people. This is about 1,000 times more, right? And these different cells that our bodies are made up of come in very different flavors, in very different cell types. Your liver cells are different from the cells on your nose. Yet all these cells live in a mutual symbiotic relationship. And if a single one of them turns selfish and forgets to pay attention to the rules of the body, you get cancer. So it is, think about it, right? A single person on the world turning selfish is a tremendous danger for the world. And yet our bodies do that. It's such a wonderful symbiosis. All organisms develop from a single cell. Right? So you have the egg cells, they become fertilized, and then during development you form a mouse, or you form this algae here, or you form this thing, I have no idea, it's probably a sea urchin or something like that. But that means that in development, the cells have to decide who they want to become. Right? They differentiate, they become specialized, and they do their job now in a particular spot in our bodies. I just took this movie.
This timing is very precise. Egg cells that have been fertilized at the same time divide and develop in almost perfect synchrony.
I don't need to talk. So you see these frog egg cells have been after fertilization, right? They divide and synchrony. Now cells become different, and you can see as the development goes along, you form the eyes. You can see the region where the eye forms. But all of these 3 eggs, right, develop in perfect synchrony. And then eventually the animal becomes too confined in that little eggshell and breaks out and is a happy tadpole. But in this process, the cells have taken on their different fates. They found their place in the body, and they have differentiated internally to become these particular specialized functions. Now, all cells have the same DNA. Right? It comes from the mom and the dad. This is the genome that every cell has. Yet in development, this blueprint is being read differently by different cell types. So they can then carry out these functions and change. And these signals that they receive from neighboring cells are being sensed, and they determine the structure of the cells. Right? Some of our nerve cells are incredibly long, up to 1 meter long, so all the way from the spinal cord down to your toe. With other cells, red blood cells are tiny. They have different behaviors. Some are static, some move around. Immune cells are highly mobile, right? They actually come running to the site of infection. And they have different fates. And the fates are programmed. The reason why we don't have webs between our fingers is because the cells that developed here were programmed to die. So that's how body shapes and organ shapes develop. And this can change, right? This is a layer of cells that's cells that illustrate here. If it gets injured, then these cells at the edge of this wound that previously were just statically sitting there, now start becoming mobile. They sense this gap, and that's how wounds repair themselves. So they start migrating into this space, and once they fill it up again, they stop dividing, they stop migrating, and everything's perfectly fine. It's just marvelous. How this all just happens. And cancer cells are different. They don't obey these rules. So these are cells, breast cells that are being grown in culture. And you see that when you grow them, normal cells, if you grow them, they form these, they arrange into these little structures that have a lumen and a duct where the milk is produced. So you can do that in the test tube. But if we look at breast cancer cells, they don't form these organized structure anymore. They don't listen to each other. They just form these awful clumps that keep on growing and growing and growing, disorganized and just take over in the cell structure. So all cells have sort of the same components. They have all these different fancy full names. But if you look at them, every cell has a nucleus, right, where the genome is housed. They have various skeletal elements, cytoskeletal elements that give them shape and structure and rigidity. They change from cell type to cell type, but they all have the same principal building components that then get modified so the cell can carry out its specific function. It is incredibly crowded and chaotic. We can draw these beautiful pictures, but if you look actually on an electron micrograph, you see that there's incredible chaos in there. And despite of this chaos, everything works. Everything comes together. Everything the cells are growing, the metabolism is working, everything is tightly balanced. So these are the challenges, right? To understand that and to try to figure out how do these cells make these miraculous combinations and of a multicellular organism. So our own work concerns this one particular compartment in the cell, which is fancy for name endoplasmic reticulum. It's basically a network of membrane tubes and things inside the cell. And this is where the components, protein components, that are the machinery with which cells sense information and carry out functions, are first assembled. Biosynthesis takes place here, and they either end up inside this organelle or they end up embedded in the membrane. And then they move from there onto the cell surface, so they can now sense other cells and transmit that information and lead to the right decisions that the cell needs to carry out. And it's this step here that is incredibly tightly controlled. So in normal cells, if you make a protein and that's not perfectly assembled, it will never leave this compartment. It will never be shown in cell surface. And it's this function then that we think is required to give the cell the proper quality control, so it can actually receive signals with an appropriate fidelity. And this is an electron micrograph here that shows a specialized cell. This is a cell from your pancreas that makes digestive enzymes and makes its own weight in enzymes that it secretes into the gut every day. So it's a specialized protein production factory. You see that this one organelle now basically just fills the whole cell compartment for this specialized. So it's a highly dedicated thing. And this is what we've been studying. How does this cell know how much of this organelle it needs? Right? And what we found over the years is that this organelle makes proteins, but if these proteins don't fold properly, then there are sensors sitting there that say, oh, we don't have enough of this organelle. So we get a feedback loop that basically tells the cell, make more of this stuff, so I can't do my job. This shows this happens in differentiation, in cell specialization, when a cell that is floating in the bone marrow like that becomes a secretory factory, makes antibodies, becomes a plasma cell, that now the structure of the cell changes dramatically, filling up with this specialized stuff. Now what has to happen in the organelle is that you make a protein. And the protein is made of a linear sequence of amino acids, the building blocks. And this is drawn to scale here, so you make a linear chain of these building blocks this long. And then this thing has to be folded up very intricately into these three-dimensional structures that then are the enzymes, the proteins, the signaling machines, and all the moving parts, bits and parts in the cell that carry out various functions. And it's this folding event. It's this folding event then that has to be highly controlled and be highly specialized. And it's only this protein that can be functional, right? Because different regions of this polypeptide chain now come together closely in space so that they form a surface with which some actions can be carried out. So how does the cell know whether a protein is folded properly or not? We have sensors sitting in the membrane of this endoplasmic reticulum organelle here. These sensors bind to unfolded proteins. And when they have bound, because there isn't enough protein folding capacity, you don't have the machinery you need, sufficient abundance. And when that is being sensed, then the signal goes to the nucleus of the cell where the genome is housed and leads to the expression of other proteins that build more of this organelle. So we have this feedback regulation here. And over the last 20 years or so, we have worked out the mechanism by which this takes place. We know the components that sit in the membrane, and basically on one, they expand the membrane so they can figure out what's going on inside the organelle, and then transmit that information across the membrane so it finally can regulate the gene expression program. And then the decision when these pathways are being upregulated are to bring the system back into balance. Right? You didn't have enough machinery, you need to make more. So you want to balance and bring the system back into homeostasis. But if this cannot be achieved, then cells make this active decision to kill themselves. Right? So, beautiful question. How does a cell, the single cell, figure out, right? Can I fix the problem I'm having, or do I have to kill myself? And again, we figured out the mechanism by which this works. And these three different branches have very different timing in their activation. They sense unfolded proteins, but then the first thing turns on and then turns off again quite rapidly, a matter of 10 hours or so. And these are the functions that are cytoprotective. They help the cell make more folding capacity. But if this cannot be achieved, then another branch that stays on much longer overwhelms this branch and leads to cell death. Right? And that's of course important because this is what we want cancer cells to do. Cancer cells evolve in wherever one cell has a mutation, and this mutation tells it, that allows it to not listen to the body anymore, but mutations are not directives. So as the genome becomes unstable in cells, they make more and more mistakes, Some of them help them grow better now, but other mistakes are just random. But they have a lot of unfolded proteins. They should die, but they don't. Right? So if we can twist this little balance there between cytoprotection and apoptosis, we can potentially do something about cancer. Here we just show you where we induce this pathway now with unfolded proteins. Here control cells, you grow very nicely, whereas in this case where the balance is working well and we induce unfolded proteins, you see that the cells shrivel up and just sort of hurts to watch them. But this is basically the sign of cell death, right? They die. This is what we need to bring back into the cancer cell. So the unfolded protein response, this is signaling network that we've been studying in health, is we think of it really as very fine control, right? A finely tuned homeostasis. But in disease, it can either be an executor that kills cells that we would rather have stay alive. And examples of that are inherited protein folding diseases. One example is a form of inherited blindness, retinitis pigmentosa. where the patients have the light sensor proteins in the retina with a mutation. So now the protein doesn't work quite well, but the cell cannot fix it because there's a mutation in it, right? So it accumulates in our little endoplasmic reticulum, in our organelle, and the cell gets frustrated. So it decides to kill itself. So the whole retina degenerates in these patients, leading to by cell death, leading to this form of blindness. We have type 2 diabetes. We are asking our beta cells in our pancreas to make ever-increasing amounts of insulin under our Western diets. And because of that, the capacity of these cells to fold these molecules get exceeded at some point, and the cells die of apoptosis. Of course, if one cell dies, the cell next door is asked to make even more insulin. And because of that, it becomes a spiral downwards of losing pancreas function. But then, as I already alluded to, this balance can also shift in the opposite direction, in that activating this pathway in the wrong way, on the cytoprotective side, we are now having a rogue cytoprotector that keeps cells alive that you would rather have die, right? The examples of that are virus infection, where many viruses use this compartment to make all the proteins they need. So if we could make some little agent that shifts this balance a little bit over into the cell death pathway, we could make potentially a very broad spectrum antiviral from which the virus could not mutate away, because we are affecting cell function, intrinsic cell function. And finally, we have cancer, where, as I already said, many cancers, all cancer cells, every cancer, has mutations, and that allows them to live in niches where they otherwise would not live. And if we can switch the balance over to the cell death pathway, we could develop potentially very new ways, very general ways of chemotherapy to help patients with cancer. So our challenge, however, is that we have a case of Dr. Jeckyll and Mr. Hyde. The UPR can be, this network can be both good and bad at the same time. And what we really have to figure out now in the lab with our compounds in disease models, whether we can come up with a way of manipulating this kind of signaling that happens in every one of our cells and actually have more benefit, do more good than harm in any disease setting, right? And that's where we're at. That's basically where we're at right now. So the goals for the next decade or so is to develop and understand small molecules that we can make, sort of drug-like molecules for each of these different branches, so we can manipulate this decision between life and death, and use these compounds then as proof of principle tools to explore whether we can find out ways of actually having effects. And I find this incredibly fascinating, right? We start very basic. We want to understand the basic principles of how these pathways work. And as we learn more about the basics, we look for clinical applications where hopefully we can do some good for mankind. So I want to close with just showing you a very detailed molecular simulation of one of our pathways, just to follow up on I didn't hear your talk beforehand, but follow up on your theme that one has to be happy in doing our work, because most people think of scientists, right, sitting there as nerds on the benches. So here is how we model our little pathway. We have these different components here, and you see that proteins inside the endoplasmic reticulum are malformed. You'll be happy. Here are our signaling, and now proteins enter, and they overwhelm the system. You see they're much more unfolded here, and they start wiggling around, and now our little sensors here become agitated, right? They modify each other, and they interact with the unfoldedness, so now they're really active. And then one of the things that we discovered is that this activated signaling molecule now acts to cleave this mRNA here, and it cleaves out this little disturbing thing, which floats away. Along comes the ligase that puts the ends back together. And it's then this messenger RNA that now, this is a ribosome here, this thing that puts this code and makes more proteins. So now it translates that mRNA and restores the conditions inside the endoplasmic. So we understand it all. You're not too unhappy. As a general philosophy, since being an explorer, you have no idea what's going on. You have no idea whether we will ever make another discovery. It is incredibly hard work because most of our conclusions are incredibly indirect. And we know 5, 10 years later, whether we learned something of significance. So in the lab, this is my board here. Never mind what's holding it up, keep hammering. These are my students.
So questions now. I have a question. So we know a lot about cells now. Why we can find a cure for cancer? What is it that we don't know? To summarize it. Sure.
What we don't know in this particular case is how cancer cells can tolerate the unfoldedness. Why do they no longer this? What allows them to make themselves? And that's different probably from one cancer to another. They've all found all sorts of different escape pathways. There are some cancers we can cure. I had cancer some 10 years ago, and I'm fine, I'm here. Had I lived 20 years earlier, I wouldn't be here. So we've come a very, very long way, and we have still a very long road ahead of us. It's basically understanding the details of what goes wrong in the programming of these cells. As I tried to illustrate in the first slide, it's a tremendous surprise that we don't all have cancer, given the number of cells we have and the number of things that can go wrong. So our bodies work amazingly well. You may have heard about this the immunotherapy that President Carter has helped him cure his cancer. It's a very similar thing. In this case, cancer cells that make defective proteins are actually seen by our immune system, and it attacks these cells that are now deviant, that are not normal, they're not perfectly folded, and kills them for us. In this case, we just need to stimulate the immune system a little bit. It doesn't, unfortunately, doesn't work in all situations.
Questions?
Okay, happy green.
Well, I asked a question before, so anyway, in nerve cells, which can't replace themselves in general, it would seem that the cost of going to apoptosis would be very, very bad. And what I know about retinitis pigmentosa is you can live decades, see okay for quite many, many years. These slightly wrong proteins don't quite hold up the normal way, and then it slowly, cell by cell, falls apart. Does that make sense in terms of your central model?
Yeah, I think retinitis pigmentosa, the severity of the disease depends on the particular mutation.
Correct.
So if you have some mutations that severely misfold the protein, it becomes very rapid progression. But in milder cases, it is much more gradual. And the gradualness doesn't come from an individual cell, but from stochastic cells that die. This is my understanding of the disease. And in nerve cells, they turn over their membrane proteins, so they replenish their membrane proteins. And if protein misfolding becomes too bad, such a neurodegenerative disease, cells do die, and it's very bad. Alzheimer's, Parkinson's, these are all protein misfolding diseases, where the misfolded state is an inducer of in the worst case, I mean, mad cow disease. These are.
Do you think there's something cumulative that makes the cells live for 20, 40 years or whatever, 60 years before they fall apart? Because it has sometimes the same misfolded protein, and it does okay,
yeah.
and then it falls apart.
Yeah. So we have mechanisms by which we degrade and recognize, turn over misfolded protein. So whatever doesn't fall in the endoplasmic reticulum gets just chewed up and added back to the basic food to make new proteins out of it. And in some cases, for some proteins, it's a very inefficient process. So a small percentage of what you make actually is perfect enough for the cells to keep. As we age, these quality control pathways go down. They don't work as well anymore. And that's why many of these folding diseases have late onset in our lives. One question back there.
Yeah, so if I understand, the idea for viral treatments would be that you target the cells infected by the virus and like move your sliding scale on this pathway so that you can encourage cell death. How do you actually detect which of the cells are infected by the virus so you don't just encourage death everywhere?
Yeah, I mean, the death wouldn't be, and that's precisely the question, right? Can we do more good than harm? Right? In this case, I would say you would kill the cells. You would deliver whatever agents you have systemically, unless you have a very localized infection, and just tweak the pathway slightly, right? So that the virus slows down. You do some harm with that, because we are affecting here a quality control system that we think is very important for every cell. But you would slow it down, hopefully enough for the immune system to take over and do the rest for you. So this would all be, this is not black and white. You would have to tune things carefully. But the surprising thing is that when we do animal models of these pathways, that in many cases we can substantially reduce them without any more effects on the organism. So I think we have some range in which we can manipulate the system.
One more question.
More question?
You said the sensors would sit on the outside of the cell and sense infections and send a signal back to the factory that's making things. How does that signal be sent? Is that electro or chemical, or how does it communicate? How do the sensors communicate?
Yeah. So these sensors are actually sitting in the membrane of the organelle themselves. So the signal they see is a chemical signal, which was in this marvelous animation I showed, is transmitted by cleaving that messenger RNA, so that white thing. So it changes in chemical composition after that, and then encodes a different signaling molecule. One more? Right?
Yeah, how does your quality control system allow prions to self-replicate? I mean, those proteins, they affect their neighbors and they replicate without being manufactured by any.
Yeah, so prions are these nasty proteins that fold right into structures so tight that they cannot undo themselves anymore. And form these incredibly, basically undigestible clumps that are not toxic for cells, such as in mad cow disease. The quality control system ignores them because they do not bind to these sensors. So they are just producing themselves. So we have no idea yet how to go about that. The best way is, I mean, Stan Prusner is here at UCSF. He got the Nobel Prize for prion diseases. And they're working actively in trying to find molecules that interfere with them ever coming together, that would destabilize these clumps, right, by allowing the supplements to develop.